Hello, thank you for joining the webinar today entitled Fatty Acid Balance, Mind the Gap, Re-examining the Evidence in Preterm Lipid Emulsions. My name is Jody Wolf, a Senior Medical Science Liaison and part of the Baxter Medical Affairs team. Before I introduce the speaker, let's review a few housekeeping items. If you have any questions, please use the Q&A button that you see at the top of your screen. We will have some time at the end of the presentation to address your questions. We will also be recording this presentation and it will be available on our medical education website. And now let's meet our speaker, Dr. Camilia Martin. Dr. Martin is a clinical professor of pediatrics at Weill Cornell Medicine and a chief science officer at the Charles H. Hood Foundation. She is nationally and internationally recognized as a leader in neonatal nutrition and metabolism with a focus on lipid and fatty acid delivery in the preterm infant. Her research spans basic, translational, and clinical investigation, examining the impact of early nutritional exposure on organ development, growth trajectories, and lifelong health. Her scholarly contributions include more than 100 publications, 4 books, 18 book chapters, and over 180 invited national and international presentations. And now I'll turn it over to Dr. Martin. Cami?
Thank you, Jody. Thank you for that introduction. And it's really a pleasure to be here today. And thank you so much to all of you for joining me. I love discussing fatty acids, but I hope to hear from you as well. And your thoughts about sort of what I present and what I've been learning over the course of my career, and also the considerations that you have when we try to implement this data to the bedside and to our infants. Here are my disclosures. So the learning objectives for today. First, I want to examine the unique fatty acid challenges that are faced by preterm infants, highlight the research, and connect those fatty acid changes after birth to short and long-term health outcomes. And then describe how our choice of lipid emulsion influences the fatty acid balance during this perinatal transition, and then initiate a forward-looking discussion on future directions, like what else can we do and study and evaluate to ultimately meet the unmet needs in lipid management of preterm infants. So let's begin by examining the unique fatty acid challenges faced by preterm infants. So what is the problem? Like, why are we talking about this topic? And why do I study this topic? Well, as you may be aware, this is a depiction of the fatty acid metabolism of fatty acids going from the essential fatty acids, as we have in linoleic acid and alpha-linolenic acid. They're called essential because we must get them from our dietary sources. We cannot endogenously make them. Linoleic acid is part of the N6 pathway that goes on to form arachidonic acid as being one of the key fatty acids. I should say that I am concentrating on arachidonic acid and docosahexaenoic acid and a little bit of linoleic acid and EPA. But there are many fatty acids, and a lot of them are very bioactive, which makes them very fun to study and how essential they are to the human biology. On the N3 pathway is alpha-linolenic acid that goes on to EPA and DHA. Why I wanted to show this slide is because we'll be discussing the fatty acid metabolism as well of these, what I call parent fatty acids, where arachidonic acid goes on to form prostaglandins, as well as thromboxanes, leukotrienes, and lipoxins. And then docosahexaenoic acid, resolvins, and neuroprotectins. And it's really important to understand those terminal mediators because it helps explain some of the biological impact we see from these parent fatty acids of ARA and DHA. So in preterm, for our preterm infants, it's important to understand fetal physiology and what was supposed to have occurred if the infant had remained in utero. And this is my favorite slide about what we know about fetal physiology and long-chain fatty acids and how important it is in development. But it also, which I'll bring up a little bit later, gives us a hint of maybe what our targets may be after a baby is born prematurely. So on the left is an example of a nice picture exemplifying the term biomagnification that's often used when we talk about fatty acid transfer from mom to infant or mom to fetus. And so what you see in this graph on the left, on the y-axis, is the circulating phospholipid in DHA percent, and on the x-axis, gestational age in weeks. Maternal fatty acids, or DHA, is in the solid green line, and the fetal is in the dotted line. And you see that late sort of third or second trimester going into the third trimester, just after 25 weeks or so, the fetal circulation of DHA and the phospholipid content or component starts to outpace that and be higher than what is in the maternal circulation. And this is why it's called biomagnification. This process is facilitated by very specific and selective transfer across the placenta. So it's not as if all these fatty acids in mom circulation gets transferred over to the fetus. It is selective for DHA and ARA, kind of telling us even more so how important these fatty acids are in fetal development. Here is in the middle slide is another example of the biomagnification when you talk about the fetal compartment to the maternal compartment. So if you look at, for the example here, just in the systemic fatty acids, you see that arachidonic acid in the fetus is about twofold higher than what we see in the mother, and DHA is also about twofold higher than we see in the mother. If you look at just on the fetal side, this is where I think you start to understand that ratio of ARA to DHA, which is very important. Think of the fatty acids of working together in an ecosystem. And so when you drive a balance towards DHA or omega-3, you're going to have that reciprocal change in the ARA and the N6 pathway, which may alter, again, their biological effect on the developing baby and the developing infant. And so when we look at the tissue compartments within the fetus, you start to see what those ratios look like. In the adipose tissue, you see that ARA is 9% to DHA of 1.6%. So a ninefold greater of ARA to DHA. You look at the systemic circulating pool, it looks like it's about 3-to-1 ARA to DHA. And then you look at the brain is about 2-to-1 with ARA outpacing that of DHA. They're both very important, but it gives an example how concentrating on just one fatty acid over another when we think about our postnatal management might start to have this ratio at an imbalance and not really drive the biological pathways that we're hoping to achieve. The slide on the right illustrates to me and is a reminder to me why the preterm infants are so vulnerable after delivery and into the early postnatal period of developing these deficits in these really critical fatty acids. I'll show you later that at birth, they actually are pretty close to what babies are when babies are born at term. And that's important because often you'll hear that, oh, because the babies are born prematurely, they're born in deficit. And I will argue that our data actually doesn't show that. They're not born in deficit, but they do acquire early deficits. And we'll talk more, but on the right is a reminder of just why that may be and why these early deficits occur. And so in this slide, it's depicting the accretion of, here is an example again of DHA in different tissue types. And yes, as we have learned historically that it's important in the brain and in the eyes and other lean tissues and organs, look at the fat stores and the accretion in fat stores, which really accelerates during that third trimester, which is often where babies and fetuses develop those large fat stores. Well, when you're born prematurely, you don't have that opportunity to build that adipose tissue. So you're not born with a reservoir of fatty acids that you can draw upon. You're immediately reliant on what's provided to you nutritionally. And you'll see, and as you may already recognize, our nutritional repletion of these fatty acids, we have a long way to go, and the babies are then born, despite being born relatively sufficient, immediately go into these postnatal deficits. And here's the timing factor. And this is, I purposely put this study back in 1992 because when I started studying this a bit after this, you always think, oh, some interesting novel findings. But this postnatal deficit that preterm infants experience is not necessarily novel and has been described before. And here's a paper in data from 1990s. This is in infants a little bit older, but we see the same pattern that we see today, even in our more immature babies, where within the first week, so again, in each of these graphs, the y-axis is the mole percent of your fatty acid, DHA, middle panel, arachidonic acid. And you see kind of what I'm talking about or was discussing earlier, that the levels are pretty replete, but then within the first, and here you see 2 weeks, I'm going to show you data that happens earlier than that, but for them, they measured then at 2 weeks, and then most of them sort of nadir out at 2 weeks. And then they sort of go into this steady state, but it's well below what should have been if the baby had remained in utero or compared to what birth levels were. So this is that deficit, that dip that occurs in these fatty acid levels, and in this study, within a couple of weeks. And then on the far right is what we see with linoleic acid, where that actually triples almost within those 2 weeks. Despite starting at about 8 mole percent, you're seeing them about 20 to 22 mole percent. So essentially what is happening is that soon after birth, the absolute levels of these fatty acids and the relationship, the ratio of these fatty acids to each other completely goes in the opposite direction that what they were seeing in utero, which is obviously optimized for fetal development. And here's the slide in 2011, and this depicting the study. It was one of my first studies that made me really become interested in what fatty acids are doing in preterm infants. And you can see the patterns are very similar. So on the y-axis, again, the DHA level is mole percent. And here we have LA and arachidonic acid levels is mole percent on the right. In the shaded area, this study, we were able to analyze in about 10 term infants what was birth levels, because I wasn't sure what to expect in preterms or what is even considered their target or where they would have been. And then so what we see in DHA, you see, again, this is what I was talking about. The preterm and term infants start very close to each other. The preterm infants aren't born in deficit, but what happens with the preterm infants longitudinally, just as I showed before, here it was within a week, you show this decline and then the steady state, but this deficit between where they should have been if born term and where they are is fairly substantial. And then on the far right, you see the same thing happening to arachidonic acid, and again, that tripling of linoleic acid. But please note, very close preterm to term, and if anything, the preterm infants with arachidonic acid was higher than that of term infants. No matter what study you look at, this pattern remains repeatable and the same, where you are born at a certain level in arachidonic acid, DHA, within days. Here, there was a study more recently that showed within 3 days, you're already starting to see that fall, and in the steady state, which fails to achieve what should have been maintaining birth levels or what had been in utero levels. So this is the issue, and this is the problem where we have these dramatic changes early after delivery in fatty acid status once born than compared to in utero levels. In our current nutritional practices, where you see that steady state and never really a return back to baseline, our nutritional practices fails to maintain those birth levels and those ratios of critical fatty acids. And conceptually, how I kind of think about this and understand the impact this is to overall development is that if you kind of divide the course, the longitudinal course of a preterm infant over the course of the NICU, you've got that early phase, that early postnatal phase. Here I have it in a week. You see that deficit occurring. And then this deficit remains throughout the remainder of the NICU. And we have to remember during that time, they're continuing ongoing development of their immune system, of gut development, brain development, eye and lung development. But now they're doing it with absolute levels and ratios completely opposite to what these organs should have seen. And then so the later convalescent stage, we start to see neonatal morbidities and in diseases such as lung disease and infection that are linked back to these deficits that have occurred earlier in their NICU course. So sometimes we see these metabolic changes in preterm infants after delivery. How do we know that these do matter for overall biology and short and long-term health outcomes? Here I just want to highlight a couple of studies. This going back to the study in 2011 where those changes were depicted by GC mass spec and whole blood fatty acid levels, and again seeing that deficit and then the tripling and excess of LA. In those infants doing the best we could as far as controlling for all other factors, every 1 unit drop in that mole percent increased the risk of lung disease by 2.5-fold. Every 1 unit increase in that LA to DHA ratio increased lung disease by 8.6-fold. Then if you looked at the sepsis, every 1 mole percent drop in arachidonic acid, there were a 40% increased risk of late onset sepsis. Every 1 mole percent increase in LA was a 25% increase in late onset sepsis. And then every 1 unit increase of the LA to DHA ratio, a 4.6 increase in late onset sepsis. And so despite controlling for all severity of illness, gestational age, we were seeing this linkage of fatty acid deficits to outcomes. But we were seeing some selectivity of how they manifested itself. And you'll see some of that starts to make sense when you look at mechanistic studies performed by using some animal models. Other groups, and there's other groups that have definitely looked at fatty acid balance and in preterm infants and its long-term significance, and also evaluating how best we can replete these fatty acids in preterm infants. Here's an example from Dr. Hellstrom's lab, where in a clinical study, they were able to show that low arachidonic acid was also linked to retinopathy of prematurity. And where you have the lower the levels, the more likely to have severe ROP. And so multiple studies showing the impact on lung, immune ontogeny, immune sort of reactivity, and ROP. So how does the choice of lipid emulsion influence the fatty acid balance during perinatal transition? And I think before we can start to discuss the different options we have, especially with regard, again, to maintenance therapy, like what would you want to start right after a preterm infant is born, we have to understand what's our target? Where do we want to be? Well, I hope you were able to start to glean some of that from the prior slides, where I do believe the fetal physiology starts to tell us that answer, where both are important, arachidonic acid and DHA. But during early fetal development, ARA biomagnification is much greater than that of DHA, and again, of ratios of 2 to 9 to 1. And this makes sense because when you start to look mechanistically and understanding what each of the fatty acids do, ARA, even more so than DHA, is really pro-development and organogenic during that fetal and early neonatal period. And often we discuss about arachidonic acid or omega-6 pathways being pro-inflammatory and often instilling caution and use of these fatty acids. I do believe that's kind of adopted or inherited from the adult literature, even though the adult literature, I think, is starting to also fine-tune exactly what we mean by pro or anti-inflammatory with regard to arachidonic acid. Because if you recall that fatty acid metabolism picture, there were multiple terminal mediators that emanate from arachidonic acid, and some of them are definitely pro-development and organogenic, such as prostaglandins and lipoxins. I'm going to show you some data that shows that ARA is a dominant fatty acid in preterm brain, and then some of the animal studies that we did to demonstrate its impact on lung development, gut development, and immune responsiveness. Here's a study back again in the early 1990s by a group that looked at the different fatty acids, and here arachidonic acid and docosahexaenoic acid in the cortex of brain across gestational age in infants that died of otherwise non-neurological conditions. And so they were able to get a nice spectrum of gestational age shown on the x-axis from early 20 weeks of gestation all the way to post-term. And then on the y-axis is the mole percent of that fatty acid. And here again, you see that arachidonic acid really is much higher than docosahexaenoic acid or DHA. And it really doesn't start to equal a 1-to-1 ratio or have a DHA in the excess of ARA until after about 37 weeks. That was my red line, trying to understand where that switch point was. And it does seem to be sort of early term and after. So again, during fetal development and before term, the brain is really relying on a much greater source of arachidonic acid than DHA. So when we saw that clinical data and we wanted to understand that mechanism more, and since lung development or chronic lung disease or bronchopulmonary dysplasia was an important outcome in those studies, we decided to look at hyperoxia-induced lung injury studies to understand how the balance of these fatty acids impact the incidence of lung injury. So we use the classic mouse model, established model of hyperoxia-induced lung injury, and this is in mouse pups, where after born, you randomize them to higher oxygen levels or room air. They continued to be dam-fed by the mothers. And then what we did is because it was difficult to provide just the substrate of DHA and ARA, and also knowing how hard it may be to change that in the preterm infant, we went straight to the terminal mediators. So we either gave Resolvin D1, which is a product of DHA, or lipoxin A4, a product of arachidonic acid. On the upper left is what happens to lungs when you are in a high oxygen environment. You have very enlarged alveoli, you have very thick septal walls. Very characteristic if you look at pathology slides of infants with bronchopulmonary dysplasia compared to the very small lung alveolar units you see there in the room air inset. When you give a Resolvin D1, you don't really change that morphology of the alveolus and the alveoli, but what you do see is that the septal wall becomes much thinner. And we did see by gene expression that Resolvin D1 and DHA was very important in reducing inflammation and reducing markers of fibrogenesis. If you gave lipoxin alone, we actually did start to see an improvement in alveoli number. And so we saw increased alveolization, but we also saw the benefit of decreased septal wall thickness. And together, here in the yellow inset, you almost have a mouse lung that's indistinguishable that of room air, despite being in 90% oxygen or greater. So this was the first time I think we saw in the literature that a derivative of arachidonic acid was essential in alveologenesis and improving alveolar development and lung development. Soon after this, another group had looked at in a guinea pig model and providing intralipid or SMOF, and they saw something very similarly. And so intralipid is sort of the has the, we'll talk about the fatty acid levels, but does have elevated LA, linoleic acid, but it has higher arachidonic acid than what we see in SMOF, despite seeing the SMOF having some ARA and DHA in it. And what we saw, or what that group saw, was also a decreased alveolar number with the infusion of this 4-component oil. Now, there wasn't any evaluation of the fatty acids there, but since it came around the same time as my study, I was thinking that it was probably because of a reduction of the pro-alveologenic terminal mediator that was derived from ARA. We went on to look at the gut development. We did this in a way in studying it in mice that you can standardize the diet, but the wild type mice will have a kind of an omega-6 profile, and the fat one mice will have an omega-3 profile. So it was a great way to look at the difference between an omega-6 diet versus an omega-3 in fetal development, in the impact of the fetus, while standardizing for diet. And when we saw that, you see by 3 days, a very clear distinction of what genes are being up or down regulated. So to me, as a physician and providing nutrition, very humbling, but important to see how nutrition matters so much in the signaling of tissues and in organ development. But importantly, what we saw is that the omega-3 dominant profiles started to have a decreased expression in innate defenses like toll-like receptors, markers for tight junction formation, markers of innate immune defense. And so there's a balance. There's a potential sacrifice when you drive one pathway over another and don't, again, keep it relatively in a balance that organ is expected to see. In the middle is the histology, and hopefully you can appreciate how all the blue goblet cells, you can see a definite decrease number on the right's the quantification, a decrease in the number of goblet cell production and mucus production as a result of that. And then you see a change in crypt villus height. And so there's almost like a stunting of that growth of the intestinal epithelium in the fat one or the omega-3 dominant. Now, this was the second study back to back where ARA or omega-6 profiles was associated with tissue growth relative and compared to its counterpart of an omega-3 dominant profile. So now we see it important in alveologenesis, and now we see it in gut development. The third time we saw it again, we weren't necessarily going after it specifically, but something we repeatedly saw where these derivatives in arachidonic acid was important in tissue growth. So I showed you lung, I showed you gut. Here's another example of gut. This is in piglets, where when we provided a ARA to DHA lipid, so this was intralipid, this was a 1-to-1 ratio of ARA to DHA, and then the far right, the purple, was a 2-to-1 ratio. We saw again, when the balance of ARA exceeds that of DHA, here at least at a 2-to-1, we saw a difference in villus height and epithelial growth. A more recent study looked at the impact of prostaglandin E2 on the recovery of the epithelium, the intestinal epithelium, in a neck development model. And again, this is a product of arachidonic acid. And you see that with the delivery of prostaglandin, also in the face of a neck inducing model or necrotizing enterocolitis inducing model, you see better survival and you see better epithelium and tissue repair. So we have to think about this during this developmental period, not just for organogenesis, but how we should be thinking about it when we want to provide nutrition after potential injury. ARA is very important for innate immune and inflammatory responses. So these studies emanated from the observation we saw earlier about the impact on sepsis and how a decrease in arachidonic acid increased the risk of late onset sepsis. These studies were done in piglets. And what we did is we provided the piglets 10 days of different lipid emulsions, whether a soybean oil emulsion, that's in red, or green, the mixed oil, 4-component oil lipid, or in blue, a fish oil containing or dominant lipid, and that was in blue. So hopefully you can see that they pretty much cluster in all those terminal mediators, or we call them oxylipins or specialized pro-resolving mediators. They come with many different names, but you can kind of see the clustering of the colors. Sometimes there's a little bit of an overlap, but there's a very distinct lipidomic profile that's induced based on the lipid emulsion you're providing. Again, should be humbling to all of us how swiftly this occurs after providing a lipid emulsion. Then the next question is, does it matter? Does it matter if we have a lipidome that's to the left, or the blue in the middle, or the green to the far right? And so at the end of the 10 days, we provided an 8-hour infusion of LPS, again, to understand that relationship we saw to sepsis. And when we did this, this was the change in that oxylipin production during LPS infusion. And interestingly, most markers did not change, but some did. But they did only in the first hour and only in the group that was receiving soybean oil. And you see this kind of reflected when we looked at one cytokine, multiple ones, but this one, IL-1 beta, where you see this response in the soybean oil, but not so much in the other oils. And then the differences kind of go away. It's really just in this first hour. So this sort of acute inflammatory response. Now, we must always ask ourselves when we do this kind of studies, is seeing a response a good thing or a bad thing? And I'm saying that this here, it's a good thing, and this is what our body is supposed to do. We're supposed to be on ready to make an appropriate attack with any bacterial invasion. You don't want to suppress that. What you don't want is that acute response to linger and become a chronic response and a chronic environment of inflammation that then goes on for tissue injury. And that is thought to be the mechanism of many diseases that we see in the NICU and during the neonatal period. Not many deaths of piglets in this study, but the few that we did have, they were mainly in those that did not receive the soybean oil, or they received the fish oil containing lipid emulsion. Finally, to end this story, just because I thought it was fascinating that this was seen in zebrafish. And so why zebrafish, and how is that possibly a good thing for comparison to babies? But apparently zebrafish are the only ones that sort of have maintained this receptor, OXER1, which is a receptor that interacts with a metabolite of the arachidonic acid pathway. You don't see it in mice and you don't see it in other mammals, but you see it in zebrafish. And so zebrafish here is a good model to study this pathway. And what they show is that arachidonic acid, the 5-oxo-EAT metabolite, interacts with the OXER1 receptor, and that interaction was critical in mounting that initial physiologic rapid response against bacterial infection. So it was just another model system, how these metabolites off of arachidonic acid really primes the host to be able to sufficiently respond to any bacterial invasion. And although we know very well the importance of DHA and its anti-inflammatory responses, again, we need to also change our language and our perceptions to understand that ARA does something very similarly. And this was another study showing that both of them were integrated parts of the inflammatory response, and both of them showed reduced pro-inflammatory cytokine of IL-6 with increasing levels of DHA or ARA. And if you go back and look at the lung model, we saw the same thing there, that both derivatives of DHA and ARA were anti-inflammatory, reducing the expression of a pro-inflammatory cytokine. So the intent of showing you much of this and from the perspective of ARA is to really understand how that fatty acid is critical in some components that have been ascribed to DHA, such as anti-inflammatory, but it adds the component of being essential for tissue growth, organogenesis, tissue repair, all important things. And then also, it's a good partner to DHA. And there's been studies to show that DHA is in fact only as good as the ARA. So I showed you a little bit already, like in the piglet study, we're moving from a 1-to-1 to a 2-to-1 ratio allowed improved villus height, and so epithelium or gut development. In humans, this was shown in two ways. One was a study looking at long-term development based on a formula composition of a 1-to-1 ratio or a 2-to-1. And it wasn't until they were at a 2-to-1 ratio that they showed approximation of developmental scores relative to that of breastfed infants. Whereas if they were at a 1-to-1 ratio, they were not only very much different from breastfed infants, but also different from the 2-to-1 ratio of ARA to DHA. I showed you early on the Hellstrom study that linked low ARA to ROP and initially had not shown that sort of independent effective DHA. But this is an article on the right showing a secondary data analysis of their data. And when they looked at the impact of DHA on retinopathy prematurity, when arachidonic acid levels were sufficient, and here they sort of was, beyond a mole percent of 8.5%, they saw that DHA then started to have a decrease odds for ROP severity. So it was very important, or it is very important in ROP, but the importance of DHA was magnified or shown when the ARA levels were sufficient, not when they were insufficient. When they were insufficient, DHA actually showed an increased odds of ROP. So I kind of like to think about it that ARA is that early fatty acid that we see very high during fetal development. That's very important for tissue growth and organogenesis and immunotogeny and repair. It's because it provides this fundamental structural foundation of tissue growth and structures, and all of that needs to be there, such as a foundation of a house, before any other kind of finishing touches by DHA can then be applied. If DHA is acting on a tissue that doesn't have the important elements present and doesn't have that structure and growth, then it's acting on nothing. But if that's in place, then DHA is allowed to shine. It's kind of how I'm starting to understand the fatty acid interaction. So there is this essentiality of arachidonic acid that's important across all of these spectrums that we have spoken about. And again, a concept that has been discussed for many years. And I just want to make sure we don't lose this concept when we think of premature infants in the NICU, and especially as we get younger and younger in those infants that we provide care to that are still undergoing tons of organogenesis and tissue growth. So when we use fatty acids to reduce inflammation, to drive these anti-inflammatory processes. And again, the intent, I completely understand why you would think about that, and the omega-3s and the DHA. But during fetal period, I think we have to be careful because it's that balance that is also very important in organogenesis. So you almost create this tug of war. So how do I take that data to start to inform what we do? So this is probably not completely reflective of today's practices. I have parenteral and enteral very distinct. Obviously, we know that we're feeding earlier and earlier enteral nutrition, which is great. And we can talk about how that impacts lipid delivery. But I am going to concentrate the next slides on how we think about the parenteral phase of lipid nutrition. So right now, there's 3 available lipid emulsions to us. There's 100% soybean oil, there's an 80% olive oil and a 20% soybean oil combo, and then there's the 4 oil, soybean oil, MCT, olive oil, and fish oil. And it's very predictive what each oil will do with regards to fatty acid or inducing fatty acid levels in infants based on their composition. So the soybean oil is 100% soybean. Soybean has a large proportion of linoleic acid, and with that, some phytosterols. The olive oil, 80% olive oil and 20% soybean oil, has less of the soybean, so less of the LA. So you would expect less of an LA rise when we saw that tripling. And because less of the soybean, less of the phytosterols. And then the 4-component oil is 30% soybean, so less than soybean, but a little bit more than the olive oil soybean. So it has kind of in the middle, the linoleic acid, and it also, because it has some components, still has the phytosterols. So olive oil is 80% olive oil. And so you'll see that expressed in sort of the oleic fatty acids. And then you have the MCT oil, the 30% MCT, medium chain, and then the fish oil, which is going to have some arachidonic acid, more than others. The DHA at 4.4, the thing we need to remember, it also has EPA, while the others do not. Again, it still has the phytosterols, but because it has more of the long-chain fatty acids, it'll have alpha tocopherol, which helps with the oxidation of long-chain fatty acids. Okay, so what happens? And I'm going to try to, sorry, 15 minutes to go. So this is what happens. Again, I said it was very predictable. So if you start with linoleic acid, soybean oil has the highest rise, with the olive, and then the 30% medium, the 30% 4-blend oil, the blue in the middle, and the olive component, less of a rise. You don't prevent the rise, but you can dial in on how much of a rise that you see. You don't want to see that tripling in the outer proportion LA to ARA in DHA. Let's go to the arachidonic acid, the fatty acid distal to the linoleic acid. So what's interesting here, again, you don't really prevent that early postnatal decline. But what you do see is that the fish oil containing, despite having arachidonic acid, is much lower than that of the soybean oil or the olive oil containing lipid emulsion. DHA, you don't prevent that decline. So we have a long way to go for all of our lipid emulsions, but you don't prevent that decline. But after that decline, you do start to see an increase in the mixed oil where you have more DHA. But here's, I think, another concerning thing outside of what happens to your ARA balance, where the EPA containing lipid emulsion, the 4-oil emulsion, has a substantial rise, much above what would be expected in a newborn infant. And I think we need to be careful of driving fatty acids that they're not even supposed to see at those kinds of levels. And EPA has many important physiological effects that have been demonstrated in adults, but driving pathways may be good for adults, but not good for preterm infants. And so when you look at the ratio of ARA to DHA, that change in the reverse direction is much greater in the fish oil containing lipid emulsion versus what we see in the other two, which relatively keeps that balance of ARA to DHA. So if I had to summarize in a report card, so to speak, on what each of these different lipid emulsions do to these critical fatty acids, this is how I would play it out, where green means you kind of maintain what we see at birth, red means it's far higher or far lower relative to the other ones, and then yellow is it's still not great. You're still having that deficit or excess, but it's less so than the other comparative lipid emulsions. And so you see the soybean oil, again, much greater LA compared to the other two. Arachidonic acid, you still see the deficit, but much greater in the mixed oils. DHA, you still see the deficit, but less so a little bit later in the mixed oil. EPA and the ratio of EPA or ARA to DHA, does not change in soybean or the olive oil lipid emulsion, but it does substantially change in the mixed oil emulsion. So when I'm choosing at the bedside and I'm thinking of the preterm infant, I am thinking of ARA as an important fatty acid to maintain again all of those structural and developmental impact it has, that it's still critical for a preterm infant to continue to develop as it goes through the NICU period, allowing DHA and other things for its maximal effect by providing us a tissue and organ development that continues to develop and has those important structures. And so I'm looking which ones have that least impact on ARA and the least impact on the ARA and DHA ratio, and probably also the least impact on that LA to ARA and DHA ratio. So I know fish oil containing emulsions have become very popular. And I often ask my colleagues, why the adoption to that without really a tremendous amount of supporting data. And just want to summarize, I hear that omega-6 is pro-inflammatory, and I hope I have shown you that I think that's a carryover myth that is pro-development, important immunity, and inflammation. They say DHA is good for neurodevelopment. It is, but you need the structural components in place for it to make its maximal effect. It's only as good if you have replete ARA and less liver injury that we can talk about, but that really hasn't panned out as well. I think there's other strategies we can do to have less PN-associated cholestasis, and we haven't seen much in the way of differences even in head-to-head trials. So in the current review of IV lipid emulsions in Cochrane, this was in 2019, I think that was the last one, they did not find any particular lipid emulsion with or without fish oil to be better than the other. So I go back to that tug of war. Then why do we want to force that tug of war if we're not getting any benefits from the fish oil containing emulsions? This was a trial from 2017. It was enteral DHA only. So it wasn't quite a lipid emulsion, but this outcome was contrary to what the hypothesis was going into that study, where they showed and they conclude that IV lipid emulsions providing DHA were at doses similar to those given in their trial as the enteral supplement, and that these IV lipid emulsions have had limited testing, and their results raised the questions about the safety of this strategy, because they showed when you drive DHA, you drive that omega-3, it actually impacted lung development. And we shouldn't have been surprised, right? Because we know that a replete or sufficient arachidonic acid is important for lipoxin and other mediators that was important for alveologenesis. So collectively, I think these findings make sense on the mechanistic studies. So no benefit, potential harm. Why are we then forcing this relationship and forcing this tug of war between trying to provide lipid emulsions that we think are going to reduce inflammation when in fact we're inadvertently impacting organogenesis? So where do we go from here? So I did mention kind of how I think about what to provide now. I think when we think about the fatty acid balance, and I think the biggest goal is just to understand what we're doing based on the physiology. Where do we think the physiology needs to be, and how can we drive that physiology closer to what is expected? But it is a moving target, and we continue to learn. So across gestational age, I do think the younger you are, the more immature. We're over here on the left side. We want this N6 environment. But as you get older, absolutely, you evolve into more of an N3 dominant requirement. And in the brain, we saw that kind of around term age and probably definitely in early infancy. So premature here, term and early childhood. What about development and disease? Well, development is definitely over here with tissue development and organogenesis. Chronic injury, though, I think, right, it's probably important to provide an N3. So I didn't talk about it much, but established liver injury and omegaven and how that's really been beneficial. It's more about that chronic inflammation, that long-standing, where you want to drive the omega-3 and DHA profiles to see if you can start to stop that chronic inflammation and that chronic injury. And there's other models of chronic injury, including brain injury, where those situations may entail providing something that's more of an N3 dominant. But we also learned in that next study that after the injury and you have to build tissue again, do you go back to these N6 derived metabolites as being important? So it's really, I think, moving forward, we have to think about it in the continuum of development and life and where we think we are in supporting normal growth and trajectories and optimizing health, but also responding to acute or chronic injury and where that puts us. But most importantly, also, let's kind of dispel any preconceived notions about what fatty acids do, target the physiology with meaningful mechanistic changes. When you read articles, don't just assume something is bad. Like I see articles saying, oh, it increases arachidonic acid, so it's bad. No, arachidonic acid has multiple functions. So don't assign that increase as bad until you actually measure the downstream effect and then determine whether or not that was an appropriate response. Moving forward, I think fatty acid studies probably have to do a little bit better in understanding the complete fatty acid profile and the composition of the diet, the dosing that we apply, standardize a bit of how we report the measures so that we can be comparable across studies, but also embrace the fact that there's many variants and single nucleotide polymorphisms that change the way the body handles fatty acids and probably accounts for the variability we see in our human populations. So we need to sort of embrace that as well. And also fatty acids are just one part of a vast lipidome. I think the entire lipidome is really fascinating and interesting for health, for development and health. So leaving some time for questions, I hope you see that fatty acids are highly bioactive and pleiotropic. We have to really emphasize the importance of our ARA while providing both. We don't have perfect practices now to maintain that early balances, but I think we do have choices to make it as good as possible. And the failure to do so has definitely been linked to neonatal morbidities. The fatty acid profiles and what is induced is definitely predictive by its composition. And here I was speaking mainly to maintenance. And then, as mentioned, did not discuss omegaven and intestinal failure, liver disease, or PNAC. Here's an acknowledgement of all the labs and the folks in the labs that have helped me with this data and learning more about fatty acids over time. There are had references on the slides, but here are the summation of the references. Here's page 1 and page 2. And I want to thank you so much for your time. There is about 5 minutes, and I would love to hear your questions.